Infecting a black ant queen involves a complex understanding of ant biology, behavior, and ecological interactions. The queen is the cornerstone of the colony, responsible for reproduction and the continuation of the ant lineage. Any attempt to infect her must be approached with scientific precision, considering the ethical implications and potential ecological impact. To begin with, it is essential to comprehend the structure and functioning of an ant colony. The black ant queen, typically larger than worker ants, resides at the center of the nest, protected by her workers. Her primary role is to lay eggs, ensuring the colony's growth and sustainability. Worker ants, sterile females, take on various roles such as foraging, nest maintenance, and caring for the queen and her brood. This hierarchical structure means that directly reaching the queen is challenging due to her protected status within the colony. Infecting the queen often involves using pathogens or parasites that can bypass the colony's defenses. One method is to introduce a pathogen that specifically targets ants, such as the entomopathogenic fungus Metarhizium anisoplii. This fungus infects ants through spores that attach to their exoskeleton, penetrate it, and grow inside the ant, ultimately killing it. To infect the queen, one could theoretically expose worker ants to the fungal spores. These workers, upon returning to the nest, can inadvertently spread the spores to other ants, including the queen, through grooming and close contact. Another approach involves using parasitic organisms like the forage fly, which specifically targets ants. The female forage fly lays eggs on the ant, and the hatching larvae burrow into the ant's body, eventually killing it. Introducing forage flies to a black ant colony could result in some of these flies targeting the queen if they manage to penetrate the inner sanctum of the nest. Chemical methods can also be employed to infect the queen indirectly. For example, bait stations containing slow-acting toxicants can be strategically placed near ant trails. Worker ants collect the bait and bring it back to the nest, sharing it with other colony members, including the queen, through trophallaxis, a process where ants exchange food and other fluids mouth to mouth. Chemicals like boric acid or insect growth regulators, IGRs, can be used in these baits. Boric acid disrupts the ant's digestive system, while IGRs interfere with the development, leading to colony collapse over time as the queen's reproductive capabilities are compromised. It is also important to consider the environmental and ethical implications of infecting ant queens. Black ants, like all insects, play a role in their ecosystems, contributing to soil aeration, organic matter decomposition, and serving as prey for other animals. Therefore, any intervention aimed at controlling or eliminating ant populations should be carried out with a clear understanding of the potential ecological consequences. Overuse or misuse of chemical agents, for example, can lead to unintended harm to non-target species and the environment. Moreover, ethical considerations should guide any scientific research or pest management practices involving living organisms. The use of pathogens, parasites, or chemical agents to infect and kill ants must be justified by a clear need, such as protecting crops from pest damage or controlling invasive species. Even then, humane and environmentally sustainable methods should be prioritized. In the context of integrated pest management, IPM, which emphasizes the use of multiple, complementary strategies to manage pest populations, infecting the ant queen would be just one component. IPM involves monitoring pest populations, understanding their biology and behavior, and using a combination of biological, chemical, cultural, and physical control methods to achieve effective and sustainable pest management. By incorporating these principles, pest control professionals can minimize the impact on non-target species and the environment while effectively managing ant populations. Infecting the black ant queen is a complex process that requires a deep understanding of ant biology, colony dynamics, and the ecological context. Various methods, including biological agents like fungi and nematodes, chemical baits, and advanced technologies like RNI, can be employed to target the queen indirectly through the worker ants. However, these methods must be used responsibly and ethically, considering the broader ecological impacts and adhering to principles of integrated pest management. By doing so, it is possible to manage ant populations effectively while minimizing harm to the environment and maintaining the balance of natural ecosystems. Thanks for watching. Subscribe for more.